हेलो बच्चों, वेलकम टू SPSVERSITY. In the previous session, we discussed about the photosynthesis pigments, photosystem, structure of chloroplast, what are the pigments involved, and the reactions involved in this. अभी क्लास में डिस्कस करेंगे इलेक्ट्रॉन ट्रांसपोर्ट इन लाइट रिएक्शन और डार्क रिएक्शन के बारे में डिस्कस करेंगे. पहले हमें डिस्कस करेंगे नॉन साइक्लिक इलेक्ट्रॉन ट्रांसपोर्ट. In the previous session, we discussed about cyclic, where electrons will start from PS1 and again reach to PS1. Here, non-cyclic means it is also called as jet scheme. So here electrons will go in not a cyclic way, in different pattern. So whenever the photons means the light energy absorbed by chlorophyll A of photosystem 2. So we'll start with photosystem 2. से non-cyclic electron transport photosystem 2 से शुरू होता है. So whenever light energy is taken by reaction center of photosystem 2, due to this electrons will come out. Electrons बाहर आते हैं chlorophyll से, A से. So these electrons will move through different electron carriers, like ferredoxin is one electron carrier. बाद में plastoquinone दूसरा electron carrier. cytochrome complex एक दूसरा electron carrier. plastocyanin. Or finally they reach to P700, that is PS1. So electrons will start its journey from PS2 to PS1. So either electrons will move from high to low concentration, that is movement of electrons is downhill, where we can see oxidation and reduction alternately. Okay. So this movement of electrons is downhill in terms of oxidation and reduction. So whenever electrons are ejected out from PS2 में दो electrons deficiency रहता है. Electron deficiency कैसे बैलेंस करता है इधर? इधर स्प्लिटिंग ऑफ वाटर, इसमें स्प्लिटिंग ऑफ वाटर होता है, दैट इज फोटोलाइसिस ऑफ वाटर. इसको क्या बोलते हैं कि फोटोलाइसिस. फोटो मीन्स सनलाइट, दैट इज लाइट. Lysis means splitting. So in the presence of sunlight, a water split होता है. So water split होने के बाद क्या आता है कि water से हाइड्रोजन आयॉन्स, इलेक्ट्रॉन्स और ऑक्सीजन. And due to this flow of electrons from in the form of downhill, there is a synthesis of ATP. और इसमें these electrons are taken by PS2. So PS2 electron deficiency is substituted. इधर non-cyclic electron transport में अब देख सकते हैं both PS1 and PS2 will participate along with PS2. PS1 also simultaneously undergo excitation. So इधर excitation होने के बाद PS1 से भी electrons are ejected, बाहर आते हैं. So ये electrons किधर जाता है? So PS1's electrons through electron carriers, they are accepted by NADP+, on which it is converted to NADPH+. So इसमें a non-cyclic electron transport में ATP और NADPH2 both synthesize होता है. उधर photolysis of water होता है और PS1, PS2 both will participate. So if you look into the non-cyclic electron transport, this is PS2. This is PS1, the reaction center. Okay. On taking light energy, so 2 electrons are ejected from chlorophyll A of PS2. So these 2 electrons are accepted by primary acceptor. And from again it reaches to plastoquinone. It is another electron carrier. So PQ means plastoquinone. फिर plastoquinone oxidize होता है and releases 2 electrons and they are taken by cytochrome complex which gets oxidized. Cytochrome complex on oxidation, again it releases 2 electrons which are taken by plastocyanin. So PC बोले तो plastocyanin. A plastocyanin से ultimately they reach to PS1. So इधर electrons P680 से शुरू होके finally वो किधर आता है P700. P700 भी excite होता है. P700 also gets excited due to these photons. इसमें से ही 2 electrons are ejected. They are taken by primary acceptor. So इसमें भी 2 electrons का deficiency. These 2 electron deficiency is substituted by electrons coming from P680. In P680, how this electron deficiency is substituted? By photolysis of water, splitting of water in the presence of light. उसको क्या बोलते हैं कि photolysis of water. A photolysis of water, one water molecule, 2 electrons, 2 protons and half H2O are released. Half O2 is released. O2 is released into atmosphere. And these 2 electrons are taken by P680. And these 2 H plus, few of them are taken by NADPH. Okay. This is how electron deficiency is substituted in P680. Okay. P700 से शुरू होने वाले electrons तो different electron carriers, that is FD means ferredoxin. So ferredoxin से और next finally they are accepted by NADPH and they form NADPH plus. Finally they form NADPH plus, which is nothing but energy currency. So इसमें नॉन साइक्लिक में PS2 और PS1 both are involved and both get excited by photons. और इधर photolysis of water takes place, that is nothing but splitting of water. और इसमें ATP और NADPH दोनों सिंथेसाइज होता है. So this is what happens in non-cyclic electron transport. So non-cyclic means again electrons will not reach to the same starting point. So electrons as a direction में they will reach to this method. So finally NAD. This is called non-cyclic. So इधर देखेंगे साइक्लिक और नॉन साइक्लिक का डिफरेंसेस क्या है. साइक्लिक में सिर्फ PS1 पार्टिसिपेट करता है. नॉन साइक्लिक में PS1 और PS2 बोथ विल पार्टिसिपेट. इन साइक्लिक में इलेक्ट्रॉन्स स्टार्ट फ्रॉम P700 अगेन रीच टू P700. इधर ऐसा नहीं. इधर इलेक्ट्रॉन्स वाटर से शुरू होके वाटर PS2 इलेक्ट्रॉन कैरियर्स PS1 फाइनली एक इधर से आता है कि NADP which is final acceptor of electrons and protons. Due to the splitting of water, oxygen is evolved as a byproduct. लेकिन साइक्लिक में ऐसा नहीं रहता. साइक्लिक में फोटोलिसिस ऑफ वाटर नहीं रहता, इसलिए ऑक्सीजन इज नॉट रियली evolved. So these are the differences between cyclic and non-cyclic. Okay. Due to this non-cyclic electron transport, that is due to splitting of water, it is nothing but oxidation of water. ऑक्सीडेशन का वाटर के वास्ते क्या होता है कि इलेक्ट्रॉन्स ऑक्सीजन और हाइड्रोजन्स भी रिलीज होता है. ये हाइड्रोजन्स they are accumulated in lumen. Lumen is the space present inside the thylakoid. thylakoid के अंदर एक स्पेस रहता है, उसको क्या बोलते हैं कि lumen. ठीक है. Due to this electron transport, इसमें क्या होता है कि lumen में high concentration of protons रहता है. And if you compare the concentration of protons with lumen and stroma, stroma में low concentration of protons रहता और lumen में high concentration of protons रहता. There is a difference in proton concentration. So a difference को क्या बोलते हैं कि proton gradient. इसको क्या बोलते हैं? Proton gradient. Due to this proton gradient, a proton motive force is created. That is the movement of protons. The movement of protons किधर से शुरू होता है? Lumen से stroma. So protons will move from high to low. लेकिन एक thylakoid membrane से प्रोटॉन्स नहीं जा सकते. They are not allowed. So किधर से जाने? They will go through this ATP synthase. So protons will move from lumen to stroma through this ATP synthase. So they are moving from high to low concentration. Whenever they are moving from high to low concentration, energy is released, and this energy is used for combining phosphate with ADP. Then you will get ATP. So इधर क्या आता है कि ATP. So a process of ATP synthesis इसको क्या बोलते हैं कि chemiosmosis. इसको क्या बोलते हैं कि chemiosmosis. A chemiosmosis, it will explain about the mechanism of एटीपी सिंथेसिस। एटीपी कैसे फॉर्म होता है? ड्यू टू द मूवमेंट ऑफ प्रोटॉन्स फ्रॉम लूमेन टू स्ट्रोमा। ये प्रोटॉन्स किधर से आता है? फोटोलाइसिस ऑफ फोटो किधर स्टोर होता है? लूमेन में स्टोर होता है। किसके द्वारा जाता है? एटीपी सिंथेस। When they move through ATP synthase, the energy is used for combining phosphate with ADP. So इसके हमें यहाँ क्या आता है कि ATP. So finally इसमें लिए तो नॉन साइक्लिक इलेक्ट्रॉन ट्रांसपोर्ट में हमें ATP, NADPH2 और ऑक्सीजन फॉर्म हो रहे। So this ATP and NADPH2 इसको क्या होता है? ATP और NADPH एक डॉट रिएक्शन यूज होता है, लाइट इंडिपेंडेंट रिएक्शन। So light independent reaction is dependent on light dependent reaction. So light dependent reaction products are used in light independent reaction, that is ATP and NADPH2. अभी हमें आएंगे डार्क रिएक्शन। A dark reaction किधर होता है? Dark reaction it will take place in stroma of the chloroplast. Stroma of the chloroplast. A dark reaction में carbon dioxide gets reduced and form starch, that is the carbohydrates. A dark reaction में दो टाइप्स रहता, C3 और C4 साइकिल। So C3 को क्या बोलते हैं कि केल्विन साइकिल। A Kelvin, a person name who discovered this C3 cycle, okay, by using, that is, carbon radioactive isotopes. इसमें C3 और C4 साइकिल्स रहता। C3 means C means कार्बन। सो पहले हमें देखेंगे C3 साइकिल में क्या होता है। What are the different reactions in C3 cycle? What are the reactions in C4 cycle? C3 में 3 reactions रहता, carboxylation, reduction और रीजनरेशन। पहला रिएक्शन कार्बोक्सिलेशन, दूसरा रिएक्शन रिडक्शन और नेक्स्ट रिएक्शन दैट इज रीजनरेशन। So carboxylation means fixation of CO2, that is addition of CO2. So this CO2 combines with a 5 carbon compound, इसका नाम RUBP। So RUBP का expanded form ribulose 1,5-bisphosphate। 5 कार्बन का कंपाउंड है और रिबुलोस 1,5-bisphosphate। इसमें 2 फॉस्फेट्स रहता, 1 on first carbon and 1 on 5th carbon. And this reaction, that is the carboxylation, is catalyzed by enzyme RUBP carboxylase। एंजाइम का नाम RUBP carboxylase। So RUBP is a 5 carbon compound, or CO2 is 1 carbon, combined it should get 6 carbons। इसमें 2 carbon compounds, 3 PGA, that is phosphoglycerate is formed। Phosphoglycerate is formed। इसमें हम डिस्कस करेंगे RUBP कार्बोक्सिलेज के बारे में। It is not one enzyme, it is having two enzymes. So enzyme का नाम क्या है कि Rubisco. C means it is carboxylase oxygenase. इसमें दो enzymes रहते हैं, carboxylase और oxygenase. When the concentration of high carbon dioxide is high, carboxylase will be activated. When the concentration of oxygen is high, oxygenase will be activated. When oxygenase is activated, it leads to photorespiration. When carboxylase is activated, it leads to the C3 cycle. So finally, carboxylation में हमें क्या आता है कि two molecules of 3-PGA, 3-phosphoglycerate आता है. क्या आता है कि 3-phosphoglycerate. ये 3-phosphoglycerate इसको क्या होता है? The next reaction is reduction. Okay. Reduction में this 3 molecules of 3-PGA को 2 molecules of 3-PGA reduction करने के लिए क्या चाहिए कि एनर्जी चाहिए, ATP और NADPH. So we require 2 molecules of ATP and 2 molecules of NADPH for reduction per CO2 molecule. So which leads to formation of glucose. So some molecules, that is tricarboxylics, some molecules are used in formation of glucose and some are sent for regeneration. All are not used for formation. इसमें कुछ molecules को regeneration A regeneration में again RUBP is formed. एक ट्राइकार्बोक्सिलिक से again क्या होता है कि RUBP फॉर्म होता है. Okay. इसमें again RUBP is formed for continuing of cycle. For this regeneration, 1 ATP is required. ATP चाहिए. ऐसे पूरा साइकिल लिया तो कार्बोक्सिलेशन, रिडक्शन, रीजनरेशन. For 1 CO2, that is when you take 1 CO2, it takes place for 1 ton. Such like if you take 6 CO2, it is required to form 6 tons of cycle. So C3 साइकिल में एक ग्लूकोज मॉलिक्यूल फॉर्म होने के लिए 6 molecules of CO2 चाहिए. So 6 molecules of CO2, 6 tons of C3 cycle, finally you'll get 1 molecule of glucose. इसमें ATP की how many ATP are used? So either for one glucose molecule formation, we require 18 ATP or 12 NADPH2 molecules. 12 NADPH. 18 ATP में लिए तो इसमें for 1 CO2 it will take 2 are used in reduction, 1 for regeneration, total 3. So for 6 CO2, 6 multiplied with 3, 18. NADPH2, 2 NADPH2 are used in reduction. So 2 multiplied with 6, 12 NADPH2. So in C3 cycle, total 18 ATP and 12 NADPH2 are required. So finally you'll get 1 glucose molecule. This is C3 cycle where the first stable compound is a 3-carbon compound, that is phosphoglycerate. अभी हम आएंगे C4 बार. C4 बोले तो 4C means carbon, 4 means 4 carbon compound. इसमें पहले compound इसमें 4 carbons रहता, इसलिए इसको क्या बोलते हैं कि C4 साइकिल. और प्लांट्स को क्या बोलते हैं कि C4 प्लांट्स. C4 प्लांट्स में एक एडाप्टेशन रहता क्या कि दे कैन सर्वाइव, दे कैन ग्रो अंडर ड्राई हॉट एनवायरनमेंट. ड्राई हॉट एनवायरनमेंट में वे भी जी सकते हैं. C4 प्लांट्स में एक इंटरेस्टिंग एनाटॉमी रहता, उसको क्या बोलते हैं कि क्रांच एनाटॉमी. क्रांच, it is term which is related to the, if we take this, इसमें large cells रहता which have number of chloroplast. ऐसा पिक्चर में देख सकते. दिस इज बंडल शीथ. A bundle sheath cells, they're arranged very compactly. Okay. A bundle sheath cells which are arranged compactly. Due to this, they are impervious to gas exchange and no intercellular spaces. Okay. So they are arranged like a ring-like fashion. So bundle sheath means which covers the vascular bundles. बंडल शीथ के सराउंडिंग में मीजोफिल सेल्स रहता विच आर वेरी लूजली अरेंज्ड. इसको क्या बोलते हैं कि मीजोफिल सेल्स विच आर वेरी लूजली अरेंज्ड where there are intercellular spaces where we can see exchange of gases, impervious to gases. ऐसा एनाटॉमी को क्या बोलते हैं कि क्रांच एनाटॉमी. C4 पाथवे में दो सेल्स चाहिए, एक बंडल शीथ सेल और मीजोफिल सेल. दोनों में C4 साइकिल होता है, मीजोफिल सेल और बंडल शीथ सेल. C3 प्लांट्स में सिर्फ एक ही सेल में C3 साइकिल होता है, लेकिन C4 प्लांट्स में C3 साइकिल और C4 साइकिल भी होता है और मीजोफिल सेल में बंडल शीथ सेल. C3 साइकिल बंडल शीथ सेल में होता है. C4 साइकिल में दोनों में रहता, C4 साइकिल एज वेल एज C3 साइकिल. Kelvin pathway is common for both C3 and C4 plants. C4 प्लांट्स में हमें चाहिए मीजोफिल सेल और बंडल शीथ सेल, दोनों में चाहिए. पहले मीजोफिल सेल में लेंगे. इस साइकिल को क्या बोलते हैं कि हैच एंड स्लैक पाथवे. The persons who discovered this cycle. A mesophil cell में the CO2 is accepted by a 3-carbon compound molecule. इसका नाम phosphoenol pyruvate. Phosphoenol pyruvate. A phosphoenol pyruvate is 3-carbon and CO2 is 1 carbon. Finally, you get a 4-carbon compound. इसमें एंजाइम रहता. This reaction is catalyzed by an enzyme PEP carboxylase. मेसोफिल सेल्स में रुबिस्को नहीं रहता, PEP carboxylase रहता. Due to this, CO2 combining with phosphoenol pyruvate will get a 4-carbon compound, named oxaloacetic acid. So, पहले reaction में CO2 with PEP phosphoenol pyruvate will It will lead to formation of oxaloacetic acid, which is a 4-carbon compound. इसलिए इसको क्या बोलते हैं कि C4 cycle. This 4-carbon compound will form into either in the next reaction it will form either malic acid or aspartic acid. So oxaloacetic acid is converted to malic acid or aspartic acid. Malic acid form है तो उसको malate formers बोलते हैं. ऐसे कुछ plants में aspartic acid form होते हैं, इसको क्या बोलते हैं? Aspartate forms. This malic acid or aspartic acid, which is formed in mesophyll, it is transported to bundle sheath cell. So पहला reaction है कि formation of oxaloacetate, बाद में formation of malic acid or oxaloacetate. Next they are transferred to bundle sheath cells. So next reaction takes place in bundle sheath cell. A bundle sheath cell में C4 acid either malic acid or aspartate, both are C4 acids. Okay. They undergo decarboxylation. क्या होता है कि decarboxylation होता है. Okay. That is release of CO2. A decarboxylation की वास्ते 1 carbon is lost to reach into form into 3 carbon molecule of pyruvic acid. ठीक है, CO2 को क्या होता है? The release हुआ CO2. This CO2 is involved in C3 cycle, takes parts in C3 cycle. It is accepted by rubies, that is RUBP. In the presence of enzyme Rubisco, normal Kelvin cycle. Okay, 3 carbon molecule pyruvic acid, इसको क्या होता है? This pyruvic acid is again transported to mesophyll cell. कहा जाता है कि mesophyll cell. So this CO2 which participates in Kelvin cycle and it will form glucose. A bundle sheath cells में Rubisco रहता, but इसमें PEP phosphonol pyruvic carboxylase नहीं रहता, इसमें सिर्फ Rubisco रहता. Mesophyll cells में quite opposite, उसमें PEP carboxylase रहता, Rubisco नहीं रहता. So this pyruvic acid again is transported to mesophyll cell. A mesophyll cell में जाने के बाद it undergoes phosphorylation. इसको क्या होता है कि phosphorylation and again it will form PEP. इसमें क्या form होता है कि PEP. If we take the C4 pathway, mesophyll cell में क्या होता, bundle sheath cell में क्या होता? Mesophyll cell में पहला reaction, that is PEP combining with PEP. Combining with PEP, it will form a 4-carbon compound. So it will form a 4-carbon compound. PEP is a 3-carbon compound. In the presence of enzyme PEP carboxylase, क्या form होता है कि oxaloacetic acid. A oxaloacetic acid which is formed, which is converted to again malic acid or aspartate, which is again transferred to bundle sheath cell. A bundle sheath cell mate will undergo decarboxylation and which CO2 is released, which is involved in C3 cycle. Pyruvic acid is formed, which is again transferred to mesophyll cell, where again it will regeneration, it will form phosphoenol pyruvate. So these are the events which takes place in C4 pathway. Okay. A C4 pathway में लिए तो सो पहला स्टेबल प्रोडक्ट है कि ऑक्सैलोएसिटिक एसिड, इसको इसलिए इसको क्या बोलते हैं, C4 पाथ। इसमें दो कार्बोक्सिलेशन रहता, पहला कार्बोक्सिलेशन मेसोफिल सेल में रहता और दूसरा कार्बोक्सिलेशन बंडल शीथ में सेल। फर्स्ट कार्बोक्सिलेशन एंड सेकंड कार्बोक्स, फर्स्ट कार्बोक्सिलेशन इन मेसोफिल सेल and the second carboxylation in bundle sheath cell. And these substances will move from mesophyll to bundle sheath, either bundle sheath to mesophyll, through plasmodesmata. They will move through plasmodesmata. Okay. So finally we get, that is, the C4 cycle is complete. C4 साइकिल में लिए तो कितना ATP चाहिए? C4 साइकिल में 30 ATP and 12 NADPH2 are required. Why this 12 ATP more are required? That is for phosphorylation of, that is, pyruvic acid to the PEP. And anyway, 12 NADPH2 it is common. 30 NADPH2 are required. So this is energy required for C4 cycle for formation of one glucose molecule for 6 CO2 molecules. Okay. And this C4 pathway, it can tolerate, that is, dry environments. लेकिन C3 में है तो इसमें वो adaptation नहीं है। C4 में रुबिस्को रहता, रुबिस्को में कार्बोक्सिलेज और ऑक्सीजनेज रहता। So when there is high temperature and high oxygen concentration in rubisco, oxygenase is activated. So oxygenase activated हो तो क्या फॉर्म होता है कि 3-PGA and phosphoglycolate. So this process is called photorespiration, which is a wasteful process where CO2 is lost actually. So photorespiration के लिए 3 cell organelles required: chloroplast, peroxisome और mitochondria. Okay. A glycolate which is formed in chloroplast, which is transferred to peroxisome. Okay, that is, which is transferred, it is converted to glycine. From glycine to, again, this glycine is transferred to mitochondria. Two glycine molecules, it will form CO2 and serine. Serine again is transferred to peroxisome. Again, it will form this, which is a wasteful process where the fixed carbon is lost in photorespiration. Due to this, C3 plants are less efficient when compared with C4 plants. C4 plants में photorespiration नहीं रहता, C3 plants में photorespiration रहता, इसलिए C4 are more efficient when compared with C3. इसमें हमें लिए तो phosphoglycolate, which is formed first, is converted to glycolite inside chloroplast. A glycolite, it will travel to peroxisome where it is converted to an amino acid glycine. इसमें glycine form होता है. A glycine is transferred to mitochondria. The 2 glycine molecules will form serine, an amino acid. Again, serine is an amino acid. Again, serine returns to peroxisome where it is converted to glycerate. Okay. A glycerate in chloroplast, again it has 3-phosphoglyceric acid, and this again can enter to Kelvin cycle. So which is considered, due to this, the fixed carbon is lost. A सिर्फ C3 plants में रहता. Due to this, they are less efficient when compared with C4 plants. Okay. So till now we discussed about C3 cycle and C4 cycle. बाद में मैं डिस्कस करेंगे फैक्टर्स अफेक्टिंग फोटोसिंथेसिस. इसमें एक्सटर्नल फैक्टर्स रहता है और इंटरनल फैक्टर्स रहता है. इंटरनल मीन्स क्लोरोफिल ले आता है, स्टोमेटा डिस्ट्रीब्यूशन. Chlorophyll, which is the amount of chlorophyll, which is an important pigment which plays an important role in photosynthesis. Okay. So a chlorophyll which is directly related to photosynthesis, amount of chlorophyll is directly related to photosynthesis, which will trap more light energy. Leaf is or anatomy. Young leaves will have more number of stomata. Like if you take the leaves, in leaves particularly if you take the distribution of stomata, number and structure of stomata, distribution of intercellular spaces, relative proportion of palisade and spongy parenchyma in the mesophyll or thickness of the cuticle. ये पूरे factors में rate of photosynthesis depend होता है. Okay. And when you come to that is external factors like carbon dioxide, sunlight, temperature, these all are external factors. Carbon dioxide is very one important limiting factor. Atmosphere में carbon dioxide का percentage, if you look into this, 0.03 or 0.04%. Till up to 0.05%, it can be increased rate of carbon, their photosynthesis. If it is more than 0.05, it may lead to damaging. C3 plants or C4 plant, they will respond to CO2 different concentration. Sunlight. Light में 3 factors. Quality of the light, light intensity and duration of light. Okay. Light intensity. The more the light intensity, the less the photos, because higher light intensity which may cause damaging effects. Okay. In light, if you take particularly the 400 to 700 nanometers of light, the photosynthesis takes place. Temperature की बारे में है तो, as all these reactions are catalyzed by enzymes, enzymes are sensitive to temperatures. And even in this C3, C4 plants will respond to different. C4 plants respond to higher temperatures and show higher rate of photosynthesis, while C3 plants can tolerate only lower rate of temperature. Okay. And when you take tropical plants, have higher temperature optimum than temperate plants. Water is another external factor, which is very important reactant in photosynthesis. When water is fully present, we can see maximum rate of photosynthesis. When water deficiency is the water stress. So water stress के वास्ते क्या होता है कि stomata will close. Stomata close हो गया तो CO2 exchange नहीं होता, CO2 availability कम होता है. Okay. इसलिए क्या होता है कि decrease in the rate of photosynthesis. Okay. So water is another important external factor which will control the rate of exchange of gases. Okay. So अभी तक हमें पढ़ा है external factors or internal factors. So finally we'll come to Blackman's law of limiting factor. So Blackman's, what it states: any process which is controlled by more than one factor, any process which is controlled by more than one factor, okay, The factor which is in limited minimal value, which will affect that process, which is in limited one. Like if you take this, the rate of photosynthesis and the light intensity. Here it will take CO2 and light. If you go on increasing the light intensity up to some extent, you can see there is an increase in rate of photosynthesis. Okay. When you keep the CO2 concentration constant, so here light is not a limiting factor, CO2 is a limiting factor. Even though if you increase light intensity, the photosynthesis rate will not increase. When it will increase? Only when CO2 concentration. Here CO2 is the limiting factor. So here the rate of photosynthesis depends on the CO2, which is a limiting factor. So here the photosynthesis, here we have taken light intensity and carbon dioxide. So this law of limiting factor is proposed by Blackman. इसमें क्या बोल रहा है कि the minimal value means the limiting factor will determine the rate of process. Okay. इसमें हमें लिए light intensity और CO2. Here CO2 is kept as a limiting factor and light intensity is unlimited. So it depends on CO2. CO2 becomes a limiting factor. So like this, the photosynthesis is affected by the external factors and internal factors. I say external, internal factors. Internal factors, chlorophyll, leaf phase and anatomy. External factors, light, carbon dioxide, water, temperature, which will all affect. Thank you all. We'll meet in the next session. Follow SPSVERSITY on the social media.